Thank you very much. And I'd like to start by thanking Felici because it's wonderful to come here. Thank you. Florence is just beautiful. It's my second visit, and this time we're spending a bit more time. So you've helped the economy as well. I think my wife is out damaging my economy at the moment. Okay. I shouldn't say that. So my brief today was to cover the state-of-the-art genetics and endometriosis. And the talk is divided into a number of sections. The latter sections I'll move very quickly, but to begin with, I'll go through in a bit more detail. But before I start with the introduction, this is really, I guess, Genetics 101. I just want to cover a few very simple concepts because when I put this talk together, I realised there's people in the room who know a lot more about this than me, and there's probably quite a few of us that don't know very much about it at all. So I'm trying to steer a course that gives a little bit to everybody. So DNA has about 3 billion loci, or points on it, where you can have different nucleotides. There's only 4 nucleotides, so at every one of those 3 billion spots, you've got one of 4 options. It's just that there's actually very little variation in nearly all of those loci. So all of the people in this room have the same nucleotides at roughly 99.9% of their DNA. They have the same nucleotides at 98% of their DNA as a chimpanzee, and they have the same nucleotides at 50% of their DNA as a banana. Okay, so during evolution, we haven't changed our DNA very much. We're only really interested in the bits that change because that's what makes us all different. So forget the 3 billion. We're talking about 10 million loci where the nucleotides can be different. And these 10 million points are called single nucleotide polymorphisms, meaning that they can have different DNA there. And that's why everyone in this room looks different, even though we're 99.9% the same. Now, very few of these polymorphisms actually change anything at all that we know about. That doesn't mean they don't, but most of them don't change the proteins that are coded for in the DNA. And even fewer of those polymorphisms are involved with diseases. However, some are, and so when we do a study called a genome-wide association study, or GWAS, we're actually looking for those small number of single nucleotide polymorphisms that confer risk for disease on us. I'm going to use things like SNP and GWAS throughout the talk. That's what we're talking about. Now, the other underlying part of the talk is that because endometriosis is more common in mothers, daughters, sisters, where the family members have it, we know there's a genetic component. And so the whole reason that people do genetic studies is to try and understand what's causing the disease, with the hope that if they understand what causes it, then you should be able to do something to prevent it or to avoid it and help and hence cure it. Okay. The first question is, are the genes that are altered influencing how the endometrium behaves? Now, the simple answer is yes, they are. And I'll just explain this study. It's most of the work I'm going to show you today is a collaboration between my group in Melbourne and Grant Montgomery's group in Queensland. So if we take a normal gene expression study, and I don't have a pointer, but in the bottom left, we've just got the normal menstrual cycle we all know about. Just above that, we've got a gene array chip, which has about 40 or 50,000 probes on it. And on the right-hand side, each column is a person and each row is a different gene. And what we can see is that gene expression changes between yellow and red, meaning low and high expression across the menstrual cycle. So I guess everyone here is comfortable with the idea that the genes in the endometrium change as the cycle changes. We do one other thing though. We take a blood sample, we get DNA from the women, and we look at all of the changing SNPs, or single nucleotide polymorphisms, in their DNA. And then So in this particular study, we took 123 women, no hormone treatment, took the endometrial biopsy, the blood sample. So we then had 15,000 probes for different endometrial gene expression and something like 8 million SNPs that we could check to see if they were different between the women. We looked for differences in gene expression based on what the SNPs looked like. So within 250,000 loci of each of those 15,000 probes, we said, are there any genes that are expressed higher or lower that correlates with a particular polymorphism? We identified about 18,000 that were significant. These expression SNPs corresponded to about 198 unique genes. So this was the first study that identified a number of genes in endometrium that you could say Mrs. Smith has different expression to Mrs. Jones because she has different, she has different single nucleotide polymorphisms which are driving that change. So if we take, just as an example, the left-hand panel there, the gene is CHRK1, ignore what it does. At the very bottom of the panel is the writing RS4902335. That says where in the 3 billion bits of DNA this single nucleotide polymorphism is. And then above that you can see whether it's homozygous for guanine or adenine. So left-hand column's GG, the heterozygotes in the middle, and the homozygote for AA is on the right. So basically, if Mrs. Smith was homozygous for GG and Mrs. Jones was homozygous for AA, they got quite different levels of gene expression. So this is the first evidence that we have in endometrium of genetic control of gene expression. So now you can say, well, what does that gene do? If you take all of those genes, they all do something, and we've got now a framework to say, well, perhaps something going on with some of these genes helps to explain why receptivity is down in this woman, why this woman has endometriosis, and so on. Moving on, even more interestingly, we found genes that showed differences in expression based on the genetics depending on the stage of the menstrual cycle. So the blue bars were samples taken in the proliferative phase and the orange bars in the secretory phase. So this gene here shows differences in expression based on whether it's in the proliferative cycle but not in the secretory cycle. So an interaction between the cycle and the genetics of the woman. This study was published last year. We've got another study that's currently submitted where we've almost doubled the size of the group to do the same sort of study, and now we're starting to see a lot more genes, over 2,000, that are regulated genetically, and we're starting to see some other issues that increase the complexity, but we're seeing genes that are regulated at a distance from where the SNP is in the DNA. So we're talking perhaps about transcription factors being regulated as well. So this provides the first evidence that we see, for example, in cancer work where they have individual genomes sequenced, individual cancers sequenced. Here, we've now got endometrial evidence for the effects of genetics on gene expression and what the implications are for the disease. So there are strong genetic effects on gene expression in endometrium, including control of gene expression levels in a menstrual cycle stage-specific manner. Okay, so we'll get to GWAS. The concept of a GWAS is very simple. If we start down, I don't have any pointer, at the bottom left-hand side, you can see the sequences are all the same apart from that one where it varies between C and T. So you take all the women with C and all the women with T, and you split them into subjects with disease of the normal population, endometriosis and non-endometriosis, and it turns out that in this example, out of 36 women with endometriosis, 14 out of 36 are the C allele and only 9 out of 36 are the T allele. So that raises a suspicion that if you have the C allele, you're at greater risk of getting endometriosis. Simple as that, except you're doing it for a lot more SNPs. Now, People started doing genome-wide association studies on endometriosis in about 2010, and there's been a number since then, 6 here. And one of the things about genome-wide association studies are that it's a numbers game. So if you look along the bottom there, 2,000, 5,000, 10,000, 20,000, that's the number of women with endometriosis in each of those studies. And up the left-hand side is the number of hits, or the number of significant points in the DNA that were identified. And so early on, the papers were only picking up about 2, then it moved to 6, and the most recent papers got 14. There's a study underway at the moment which Krina here is heading up, but involves nearly everybody in the world that's involved in endometriosis research, which aims to get over 60,000 cases and over 700,000 controls, and this, we think, will probably more than double the number of hits, genome-wide association hits for endometriosis. This diagram, I wish I had a pointer, but round the middle bit of the diagram, so round the middle here are the chromosomes, and round here are the SNPs, which show some evidence of being related to endometriosis. I have 2. So at the moment we have 14 loci that are statistically significant, and 1 on chromosome 2, 1 on chromosome 1, and 1 on chromosome 12 here. I'm going to talk a bit more about, because there's a number of complexities about these genetic associations. But at this stage, what we can say is that all of these points in the DNA do something to increase a woman's risk of having endometriosis, but we don't know what, and that's the challenge in front of us. So, and the other thing is that whilst we know that endometriosis is about 50% risk is due to genetic factors, all the work to date only explains about 2% of that risk. So there's a lot of risk that we haven't identified. We're at the start of the journey, not the end. So I'm going to talk now about those 3 loci that I identified before. The first one was the chromosome 1, which was a number of genes involved. This was just the cover of Human Molecular Genetics, published in 2016. But if we look at this is called a Manhattan plot. What we have along here, remember the 3 billion nucleotides. We've got about 1 million of them spread out in a line here. And we've looked at, now we've looked at nearly 17,000 women with endometriosis, and we know that there's a number of single nucleotide polymorphisms in this region here which increase the risk for endometriosis. When we look at the genes that are coded by that DNA, there's 3 of them. The first 3, not 2 together. The first one was Wnt4, which has a very significant role in endometrial development and stromal cell differentiation. So the minute that came up, everyone said, wow, it's got to be Wnt4. The second one is HSPC153, or link 00339, which is a long non-coding RNA. It doesn't have a product. Nobody liked that because we don't know what it does. It's a non-coding. So we can sort of talk about it, but got ignored. And then there's CDC42, which does lots of things, which is a little bit complicated, so that got ignored as well. So everyone was pretty confident that Wnt was important. When you then look at, this is a map again of women. We'll start at the bottom here for the link long non-coding RNA, that women that are homozygous for one allele have low production, homozygous for the other allele higher production, but there's a lot of crossover. Nonetheless, there's a significant difference here. This is in endometrium, and this is in blood, where we had much bigger numbers. Same for CDC42. It goes in the opposite direction, but there's a significant regulation of gene expression by the risk SNPs. There is nothing for Wnt4 at all, no change at all in those women. And so, in conclusion, what the study showed, that there's a block of risk SNPs on chromosome 1 that spans 3 genes. The SNP is actually located within the Wnt4 gene, not in a part that's expressed, but within an intron. But there's the SNP links back to the other two genes nearby and has no effect on Wnt4. So just because the geneticists tell you that there's genes near to the genome-wide association point doesn't mean that that gene has any role to play in the disease. Unraveling which genes are doing or having the effect on the endometriosis disease is complex, is horribly complex. So the results of the analysis strongly implicate variation in expression of CDC42 and link in endometriosis risk, but they do not provide any evidence at all that Wnt4 is involved. A lot more work's required. This is the only slide I'm going to put on GREB1 because this is one of the hits that actually makes a lot of sense and it all fits together quite well. So GREB1 is an estrogen-regulated gene. It was identified in breast cancer cells. If you knock down GREB1, it inhibits estrogen effects. So we know that if we shut down estrogen effects, we shut down endometriosis. It's plausible, it makes sense. Estrogen receptor transcription involves about 100 cofactors, so repressors and other proteins that modulate the effects of how the estrogen receptor works. So GREB1 is one of the most important of these, functions as an essential component of the estrogen receptor transcription complex. And its expression is altered in women that have different genotypes at this locus. So GREB1 fits nicely into what we think should be a gene that's, or it's plausible that it behaves in a way that influences endometriosis. Here's the third gene I'm going to talk about. So I've told you one candidate for which there was no evidence. Another candidate that fits well. This is a third one, which is another story, Vizatin. Again, this is the DNA laid out, and you can see that a number of SNPs influence VZT expression, and they're the same SNPs that increase the risk for endometriosis. So it's a good candidate. It's just, and there's a nice change in expression depending on the genetic makeup of the woman. That's all fine. But what does Vizatin actually do? It's a protein that sits in adherent cells junctions. So these are the things that make cells stick together. So it creates tension between cell junctions of cytoskeleton to strengthen cell-cell adhesion. And it's completely unclear why this should have any influence at all on endometriosis. And the at-risk allele actually increases expression. So it should make the epithelial cells stick together better. That's about the best we can say from the biology. So when we look at immunohistochemistry, it's more in glands. Well, that makes sense because adherence junctions are in glands. But it's also in all the other cells, and it shouldn't be because there's no adherence junctions in them. So the genetics has led us to look at a gene which has no history in endometrial biology, and yet it appears to be linked to increased risk for endometriosis. So what does it mean? There's no published data on the function of VZT in endometrium. That's not quite true. We've published one paper now. VZT functions in other tissues are related to the roles in the junction. So if you actually do a knockout mouse with VZT, the blastocyst doesn't implant because it falls apart. The cells can't stick together. So it causes infertility, but not really for an endometrial reason. And there's a lot of work to be done if we're going to understand the role of VZT in endometriosis. So I'm now, and I guess the other thing about all of the genetics work is that it's great when a lab produces a list of possible genes, but until another lab somewhere else produces a list with the same genes on it, you should always be suspicious, or at least cautious. These studies require, there's a lot of possibility for chance to be playing a role in these studies. So this is actually from Grant's lab again, but doing a different method, looking at SNPs that, using a chip that just looks at SNPs that modify protein. This is just published very recently, I think September. And interestingly, GREB1 came up in this study. Not much else did, in fact. So again, just a little hint that GREB1 might be one of the more important ones. So I haven't got a clock in front of me.
Right, 2 minutes left.
2 minutes. So I've very close to the end now. Just a couple of other important things that have happened. Rare alleles. So all that I've been talking about at the moment involves things that are in the whole population, but there are rare mutations, and I'm going to talk, there's two abstracts. So Ken Ward's group from Salt Lake City, these are abstracts from the SRI meeting, so March, only presented a few weeks ago. One study, 1,019 women with endo, they looked at the DNA around the 16 GWAS sites that I talked about. They actually found a number of rare genetic variants that alter protein coding. So these would be like one woman out of the whole group. 5 mutations had high odds ratios for endometriosis. So a slightly different study, a different approach, but they found evidence that some of the genes near to the GWAS sites, if you mutate them, it statistically increases the risk of endometriosis. In fact, they did the opposite. They took women with endometriosis and said, is there any increased incidence of mutations in any of these genes? So that, I think, is quite an interesting study. This study blew me away. This was, again, from Ken Ward, a database of 32 million ancestors of the current Utah population. So they have very good genetic databases. He has identified 218 surgically affected women related through 19 generations to a single founder born in 1508 in England. Now, apart from just the database, which is pretty impressive, and the fact that obviously endometriosis wasn't known for most of the time that this study was going on, this actually suggests an autosomal dominant gene for endometriosis with a high but incomplete penetrance. That would be amazing if they found it. They're looking for it. Whether that is the right explanation, I don't know. But again, it potentially opens the door to finding avenues for understanding what the mechanisms that cause endometriosis are. Somatic mutations, I couldn't give the talk today without mentioning this paper, which was published again since the last meeting. In this study, they analysed deep infiltrating lesions from 27 patients. They sequenced them, and they found that in 19 of the 24 there were mutations, and that in 5 of the 24 the mutations were similar to mutations that are known to drive cancer. So again, statistically, this suggests that, so these are not germline mutations, they aren't through the whole body, they're just in the lesion, possibly in the endometrium before, we don't know. But again, evidence that mutations and genetic differences at a local level can lead to an increase in endometriosis. Finally, epigenetics. I only have one slide on epigenetics because this gets really difficult. Epigenetics can be broadly defined as changes in gene function that do not involve changes in DNA. So we have these 3 billion loci, but then they all get regulated by lots of different mechanisms. So the cell turns different bits of the DNA on and off and makes things happen and not happen, such that, so things like methylation, hydroxymethylation, demethylation, acetylation, etc., influences how the strand of DNA is folded and what genes are expressed and not expressed. This is the next big frontier. This big, I'm part with Krina and headed by Linda Giudice and Stacey, part of a big grant looking at methylation in endometrium. Whether this will be the answer or not, I don't know, but it's certainly the next big frontier. So extensive crosstalk between different epigenetic layers maintains and regulates complex transitional networks in cells. Conclusions. There is a huge amount of complex data. I think one of the biggest things at a practical level is that you need to have big studies, because by the time you've stratified for genotype, menstrual cycle stage, cell type, so I didn't say, but the somatic mutations were all in the epithelium, not in the stroma, so you have to think about different cell types. And then we get to clinical data, so where the disease is, the symptoms, and so on, and environmental influences. And the functional studies are challenging because we don't have good animal models. Ultimately, we're going to start to define biological processes that increase risk for endometriosis. I think the fact I'm giving this talk, whereas I know, and a lot of us have been in the field for a long time, 10 years ago, this talk would have just not been possible. So there is progress being made, but there's still a lot to do. Finally, I've talked about our two groups. There's a lot of other people involved in the work I presented, but these are, I guess, the core groups in Melbourne and Queensland that have been doing this work. Thank you.
Thank you very much, Peter, for this wonderful overview. There's time for one or two short questions.
So one question is about the group of women that you presented in one of your slides, showing that there are stop mutations in several genes. Are there heterozygous or homozygous mutations?
So the mutation studies weren't ours. You mean the ones from...
When the analysis of the candidate genes allowed to discover mutations in GREB1, FN1, and so on, and you spoke about stop mutations, stop codons, right? And my question is simple. Are there homozygous or recessive mutations? I mean recessive or dominant?
So the mutation work's not ours. I don't know. Are you talking about the somatic mutation? No.
Yes. Okay. I think so. I think so. There's one of your slide. You remember? You know what I mean? Or
yeah. The rare mutations.
Yes, the rare mutations.
So were they
they're coding. Yeah. They influence coding. So
because if there is a stop mutation in only one allele, it means that the other allele is functional, and it's a dose problem.
As I said, they're not our studies, but I assume they're homozygous.
It's Charles TB from Paris. When you found some association between SNPs and endometriosis, and when the GWAS studies also, when some genes went out, did you find a correlation with the phenotype? When you found something, do you correlate the abnormalities that you found to a certain phenotype, or you didn't have the data?
Not yet. It's a very good question. We've already had one screen to look for, with our database so far, if there's any evidence between deep infiltrating, superficial and ovarian, and any sort of genetic profile. We don't see that yet, but we really need more numbers. So when I said at the end we need big numbers to stratify, as soon as you split into 3 groups, you haven't got enough numbers. So it's a very good question. I think everyone would like to know the answer to that question.
You didn't have family endometriosis in the same family? You didn't have cases in your studies?
Yes, but not enough to split up like you're saying. It will happen.
Thank you, Professor Rogers. So let's move to the next speaker. We'll try to keep this session perfectly on time, as if we were not in Italy. And so the next speaker is Daniel Weimann from France, Basic Science.
So first of all, I of course want to thank the organizers, Dr. Petralia and Dr. Chapon, who have invited me. It's a great pleasure and great honor to be here. And as you know, pedagogy is based on repetition. So there will be some repetition compared to what we heard before. And I hope, fortunately, Dr. Rogers left me some holes that I'm going to try to fill. And also the symbol of the meeting about this painting, a big thanks, because it's one of my favorite paintings in all the paintings of the world. Thank you very much. So you are probably well aware about the theory of endometriosis. There are 3 major theories. And in fact, one issue about what we are doing in basic science will be to try to understand which theory is really accurate, or maybe it's a mix of the different theories. Mostly people think about regurgitation as being the major issue for, I think, 80 or 90% of the person. This is the way endometriosis develops. But maybe with all the progress in genetics and epigenetics, we'll be able really to solve, in fact, this question. It has been spoken about genetic of endometriosis, so I'm going to say briefly some things that maybe have not been clearly or maybe enough explained for the audience, or maybe at least it's good to tell them twice. One is, how do you calculate heritability? Which is a very important question. How do you know that a disease is based on genetics, and how much of genetics is involved? you can have candidate genes when you have an idea about how the disease is evolving and what are the causes of the disease. Then you can sequence, for instance, genes that you think will be interesting in the frame of the disease. And in the last years, of course, there have been genome-wide approaches, as were presented before, either in families or in populations. And this is based in terms of theory on two different ways of seeing the things. About heritability, this is the first point. Heritability, in fact, is a ratio between the variance due to genes and the variance due to phenotype. What you observe, in fact, is only the phenotype. You can only estimate this. So the question is how to have this. And for this, you are obliged to work with individuals that are apparented, either sisters or monozygotic twins. And like this, you can estimate, in fact, what is the way of the phenotypic variants which are associated with these genetic commonalities that they share together. And in fact, an interesting point is that there have been several works, but I have picked two of them that estimate the variance of endometriosis. One is from 1999, and the other one from 2015, this one and this one. And you see that the numbers are quite similar. So roughly, it is estimated that genetics explained about 50% of the phenotype of endometriosis. And the question is now, with all the genes that we found, how much of this 50% do we explain? I'm not going to speak more about that. It's a principle of doing a genome-wide association study with some different ideas when you speak about families and population, but the idea is to try to associate the character that you are interested in with markers that are spread throughout the genome. And like this, you can find regions or genes where the markers are present, the SNP markers, and I've picked two important papers, one on a Japanese team, from a Japanese team, and the other one from a more international team, and they identify several SNPs, several of these polymorphisms that are associated with endometriosis. But one thing that was not mentioned before, quite important, I think, is the odor ratio. You see here, the odor ratio are always comprised between 1.1 and 1.4, meaning that the risk of each of these variants is increased of 40%. Meaning that if a woman, let's say the risk of endometriosis is 10%, when a woman has this variant, then her risk to have endometriosis is 14%. Okay? So it's not a lot, in fact. As has been said before, several genes have been identified, and the statistics behind is very strong. I mean, the identification of this gene is sure, and that they have been found by different studies independently. But as I said, the problem is that despite the fact that the statistics is very strong, they explain quite little of the phenotype. Today, in fact, I'm not going to go into the details of the genes, but only to say that, in fact, when you look at their function, in fact, you can find some links with what we think about the physiopathology of endometriosis, like proliferation in the uterine part, cell cycle, growth regulation with estrogen dependence, extracellular matrix, adherence. All this makes sense, and inflammation, of course, makes sense in terms of what you think about the pathophysiology of endometriosis. The most recent studies has been, in fact, the idea is to take all together all the individuals that were used and to do a meta-analysis that was published in Nature Communication last year, and they identified like this. They confirm 9 of these SNPs, and they identify 5 additional SNPs. And another study that was published the same year resulted in some others. And today, as has been said before, there are about 16 loci, less than 20, that are in fact linked to endometriosis and identified by GWAS. But again, you see that the relative risks are quite low. They are very significant. They are for sure associated with endometriosis, but they explain not that much. And in fact, when you combine all of them together, probably they don't explain, as has been said by Peter Rogers before, more than 2 or 3% of the genetic variability, which is itself much higher. Meaning that there are many, many genes that have very, very small effects. And this is not like QTL, like quantitative trait loci, where generally each gene explains 20% of the variation. So this is a complexity of genetics, and raises really the question, if you want to be honest, on how much investment in genetics will help in understanding endometriosis and helping to cure and to predict endometriosis. Now, in this 50% left of variability, phenotypic variability, a big part is played by the interplay between the genetics and the environment, which is in fact the explanation of the word epigenetics, at least one of the definitions of the word epigenetics. And for sure, this plays in endometriosis. This has been said long ago, in 2009, by this paper by Dr. Guo, who is here, I think. And in fact, of course, there's not only epigenetics, but in fact, this review showed that many genes are involved, more than the one that have been found by GWAS, and they are also impacted by epigenetic regulation. About epigenetics, just to show you this picture, how works epigenetics? Epigenetics is in fact the way of how genes are regulated. This is heritable from one cell generation to the next cell generation, but doesn't involve change in the DNA sequence. The DNA sequence stays the same, remains the same. And you see there are some modification, epigenetic modification, which are these. It's methylation of specific bases of DNA. And generally, it was assumed that when the DNA is methylated, then it tends to be the extinction of the genes which are around. And the most important issue in epigenetics are modification of the histones. The histones are making a core called the nucleosome, made of 8 proteins, and you see each histone has a protruding end, the end terminal, and this protruding end can be, in fact, the target of histone modificators. And this will be modified strongly by a lot of chemical modifications that will influence, in fact, the way the chromatin will be accessible, either open or closed. And this is playing together. And I didn't mention in this picture, there is no example of small RNA or non-coding RNA, but they also play a role in this regulation. Conrad Wellington, it was in the 50s, coined the idea of epigenetic. And he said that, in fact, the genes generate a landscape, and along this landscape, the cells can differentiate, and they fall like in a potential well here, and they stay here. In some specific cases, we can transdifferentiate from one type of cell to another type of cell, but generally it's difficult. And epigenetics is what makes that the cells which have the same genetic program will in fact differentiate in various types of cells and will be also modified in context of disease compared to a healthy situation. And as I said, there are 3 ways of doing that: the histone code, DNA methylation, hydroxymethylation, demethylation, and non-coding RNAs. About the histone codes, which is in fact the less studied thing. I started by this, and you see here the nucleosome by a plane view, let's say, from plane. And you see the tails of the histones that are protruding and that are modified. And it's a huge complexity of modification. There are at least now 80 different modifications of the end terminal of the histone and the lysines. But some of them, maybe, are associated rather to opening the chromatin and some rather to closing the chromatin. One very known is H3 K4 trimethyl, which is opening the associated to opening chromatin, and H3K27. When I say K4 or K27, it's the lysine number 4 of the N terminus or the lysine number 27. And according to the position of this trimethylation, for instance, you will have an opening or closing of chromatin, which will make that the genes around will be either active or inactive. In endometriosis, this has been the study. I think there's roughly one team that is studying that. It's a team of Idalis Flores in Puerto Rico, and they studied by immunohistochemistry mainly the mark of histone modifications, and one of them, as I said, is H3K27. So it means that the lysine 27 of the H3 histone is methylated 3 times in this case, and she could show, in fact, that according to the fact that you look at the stroma of the glands, then you can have difference, and these differences, these are the lesions, this is the endometrium, are also different according to the fact that you are in a proliferative or secretory endometrium. So, of course, again, there's an influence of a cycle. H3K27 trimethyl is in fact made by this protein called enhancer of zest, EZH2. And now they are working on that, and a very recent paper was published in 2018 about that. So EZH2 belongs to a complex called PRC2, which has been found in Drosophila, and this complex tends to close the chromatin, to silence the chromatin. And in fact, it does that by modification of the histone, like H3K27 trimethyl, for instance, and also by methylation of the DNA. So it has a double role in this. And so what they show, in fact, in this paper, published in Biology of Reproduction, is that in the lesions, the HZH2 is strongly expressed in the lesions and much less, in fact, in the endometrium, but there are some differences according to the fact that you are patient here or patients or control, and according to the fact that you look at the proliferative or secretory phase of the endometrium. They also did a ChIP-seq analysis to show that some promoters of genes that are important in endometriosis are in fact altered in terms of binding of in the presence of H3K27-methyl-3, and they also show that this has an impact in the function of cells in a cell culture experiment. This is an example of a scratch experiment, and you see in fact that the reparation of a scratch is different according to the fact that you are dealing with endometrial epithelial cells or endometriotic epithelial cells. There are differences that have been shown, and then in the paper they show some other differences. So this is something very complex, but I think it's something important for the future. About DNA methylation now, so we spoke a lot about that because it's easier, in fact, to study DNA methylation than other marks, and it has been studied a lot, and I'm going to present some of the questions. One question is, what is the link between DNA methylation and gene expression? And as I said, often it was considered that when a gene is methylated, it's downregulated. In fact, this is an example of a paper published in another context in Cell. Here you have expression of genes, and here you have a methylation. And you see that there is no correlation at all. And this makes that people move to a new paradigm. They say that, okay, it depends where occurs the methylation. When you methylate the promoter, then the gene tends to be silenced. But on the contrary, when the exons, the body of a gene is methylated, then in fact, it's the contrary. The gene is highly expressed. But also, it raises the question of what happens when both are demethylated or both are methylated. And in fact, when you look at this a bit further, in this example of a paper, they did this, but you have to go to the supplemental table number 6 to find it, because people are reluctant to say that, in fact, they don't know what happened. When you look at the promoter and gene body methylation in links with expression, you see that you can find methylation with and without gene expression. So it means that, in fact, the link between methylation of DNA and expression has to be studied by a case-per-case basis. You cannot really predict what happened. It depends on the context and on many things that have to be studied. Despite that, a few years ago, we analyzed DNA methylation by a technique called MEDIP chip. So it means that we immunoprecipitate precipitated methylated DNA and put it on promoter array. And with this technique, we did find some promoter that were differentially methylated. But again, when you look at this methylation highly methylated here, or low level of methylation here, in terms of gene expression, you don't find a correlation. You can have all the cases are possible. This can be explained in some cases. An example that I give here, you have one region which is methylated, but belongs in fact to the promoter of two genes that are going in opposite directions, and one is upregulated and the other is downregulated in the lesion. So it means that the same demethylation will be associated with something different in terms of gene expression in two genes that are nearby. Dyson published some years ago now a paper that used a microarray, a sort of array of methylation, allowing to interrogate now at this moment about 500,000 CpG that are methylated, and he found a lot of genes that were in fact abnormally methylated in endometriotic tissue. We did the same thing, and an interesting point is that these are our results on the same gene than the paper of Dyson, and you see that it's incredibly similar. It's very different to what we found with microRNA. We'll speak about that a bit later. But you see that each CpG is deregulated the same way in our lab and the lab of Dyson. And the lab are not in the same continent, and we are not studying the same patients, and we find CpG per CpG the same modification, which is quite intriguing to see how much, in fact, this mechanism seems to be conserved from one lab to the other. The major conclusion of the Dyson paper was, in fact, that methylation can explain, for instance, an abnormal balance between GATA2 and GATA6, two different transcription factors that are involved, in particular, in early development and steroidogenesis, and they could show that a shift towards GATA6 in endometriosis is associated with a lot of abnormal responses linked to endometriosis. Another interesting thing is when we compare our data and their data, in fact, you see different genes that are in common, and generally, they behave the same way. So we use different techniques, and still, the significance of a correlation between the two sets of data is very high. So again, it militates in giving the idea that methylation is something which is very well conserved and very robust, in fact, when it's analyzed. And there were some other studies, like this one, that identify some other genes candidate for that are differentially methylated. Now, Peter just spoke about this paper, very interesting one, showing that non-coding RNA have an impact in terms of epigenetic regulation, certainly. And in fact, non-coding RNAs, I'm not going to speak about them because they are, in fact, the topic of one of the presentations to come, so I'm not going to speak about that. And I'm going to move directly to some messages. The first is that I think that the histone code is a novel field of investigation in endometriosis, which is, for instance, really at the starting point, and there are a lot of things to be found that could really be meaningful in terms of understanding the pathophysiology. Non-coding RNAs are very interesting because we know, in fact, how they work by inhibiting transcription and translation, but also they can be very interesting non-invasive biomarkers. The problem is reproducibility, and the people that follow this subject know that, in fact, there are hardly 2 studies that found the same panel of microRNA differentially regulated in endometriosis. And DNA methylation, as I said, is something which is very robust. Now there are several studies that show, in fact, the same thing. And in fact, the genes that are identified are relevant in terms of hormonal regulation, steroid production, invasion, and proliferation. So not to be too long, I just conclude here. And I want to say that this work has been really, let's say, started by Charles Chaperon, that really put me in the endometriosis field, in one of my subjects. And like this, we have now a network in Cochin Hospital in Paris of researchers that are involved in this topic and trying to make things better for patients and make things better for science as well. Thank you very much for your attention.
Excellent talk, Hamama from France. So you did mention in your take-home message is there is an association between DNA methylation profile and endometriosis physiology inside relevant certain genes. My question is, if you're integrating female age, your conclusion is still true?
We didn't try, to be honest. I think, in fact, what happens in our experiment and also in the Dyson experiment, in fact, we compare from the same patient the tissue which is endometriotic compared to the normal atopic uterus. So it means that probably the age is normalized, but we didn't study it, and maybe there are some things that we could check. We didn't do that.
It's because we know that female age affects
Yes.
methylation profile.
Yes.
So for
Yes.
this reason, we have to integrate this one.
Yeah, the question is it true for uterus? Some people say that
Correct.
uterus don't age.
Correct.
But I know it's probably an exaggeration. So yeah, you are right. When you study lymphocytes and methylation lymphocyte, according to age, you have some differences. In the uterine tissue, maybe, but I don't know. In fact, what I can say is that the patients that we looked at were between the classical age where they can be operated between, I would say, 25 to 45 years.
I wanted to ask you whether the findings of abnormal methylation were found in the different sites of endometriosis in the same patient, or it can differ?
Okay, so in this case, for the genome-wide studies that we did and by our method, and it was endometrioma, so it was ovarian endometriosis in this case, and we didn't study other locations. It's really compare only with the lesion which was on the ovary and the uterus.
Daniel, I have a very quick question, and it's slightly related to that as well. I was really intrigued and heartened to see that the DNA methylation patterns that you found in whole endometrium, so whole tissue, were very similar to the Dyson study done in stromal cells. And of course, one thing that as scientists we often hear is that, well, at a tissue level, there's a cellular heterogeneity, you can't do this, you have to look at cell subtypes, which for a lot of outcomes may be true, but what is your view on that?
So it was striking for us. It was striking for us. So when you look at really genes from the Dyson study and we did use about the same tool. It's an Illumina tool, but the patients were different, and it was striking how much it is similar. And so it means that there are probably pathophysiological mechanisms that are so characteristic of a lesion that, in fact, wherever you look, and in fact, even more, in the Dyson paper, we use directly the tissue. They purify stromal cells. So our is less pure, in fact, but the results are exactly the same and everywhere. It's really striking, really striking. And I guess that there are some really basic mechanisms that are strongly impacted in terms of meditation, probably due to the fact that it's a long-term disease, in fact. So with years, the things becomes really, let's say, strong and hard, like rock, I would say. And
Yeah, very, very encouraging from that point of view. Thank you
Okay.
very much,
Thank you.
Dr. Weiner. So our next speaker is Professor Hugh Taylor from the University of Yale in the US, who's going to talk about the future directions in this field.
So first, I'd like to thank Dr. Chaperon, Dr. Petraglia for inviting me here today. What a wonderful meeting. This morning, I gave a lecture at one of the sponsored symposia on the effect of endometriosis on a woman's life trajectory, that it was more than just a few painful lesions in the pelvis. I think this lecture is going to very much parallel that. I'm going to talk about how endometriosis in a more molecular and biologic sense affects multiple organ systems and isn't isolated to the few lesions that we see in the pelvis. That cell trafficking and inter-organ communication in endometrium and endometriosis play an important role in this disease. And hopefully at the end of this lecture, I'll have you convinced that migration of cells throughout the body and other signaling molecules such as microRNAs make this really, truly a systemic whole body disease. I think we've always thought of endometriosis as a disease of cell trafficking. That we know, since the days of Samson, that endometriosis mostly arises through retrograde menstruation, that cells move from the uterus into the peritoneal cavity. But this doesn't explain lesions outside of the peritoneal cavity. It doesn't explain those lesions that are, for example, in the lung, the brain, other places that we find endometriosis. And it certainly doesn't explain endometriosis in the old days we used to find in men who were treated with high doses of estrogen for prostate cancer. So endometriosis must have other etiologies. About 15 years ago, we proposed that stem cells, for example, stem cells from bone marrow, could contribute to endometrium and endometriosis. The bone marrow has not only the hematopoietic stem cells that are used for bone marrow transplant for hematologic malignancies, but also has these multipotent mesenchymal stem cells that can give rise to these solid organs. And we reasoned that if it could give rise to all of these other organs in adults, that certainly something with a rapid turnover like endometrium, certainly it should be contributing there as well. And we did some animal experiments where we transplanted bone marrow from male to female mice and then simply looked at for Y chromosome in the endometrium. And some of the results are shown here. That the first A and B are positive and negative control, but down in C and D, the bottom two panels, you can see a Y chromosome signal in red, Y chromosome fish, in endometrial cells, and we looked at various markers to exclude that these were not leukocytes, they're CD45 negative, they express things like cytokeratin to prove there really are endometrial epithelial cells. So it looked like stem cells could contribute, and we also found that the same was true in humans. We looked at a few women who had whole body irradiation, chemotherapy, bone marrow transplant, and bone marrow transplant with an HLA mismatch. And we looked at the mismatch HLA, which you can see here stained in brown. We can see endometrial epithelial cells and stromal cells that are arising from the bone marrow donor. And what promotes this? Is this something that populates the endometrium in every menstrual cycle? Well, no. To make a long story short of all the different things that we looked at, really injury is what promotes the migration, recruitment, and engraftment of stem cells into the uterus. When we took our same model where we took male bone marrow, transplanted to females, injured the uterus. Here we use ischemia reperfusion injury. You recruit about twice the number of stem cells into the stroma in the injury model than in the uninjured model. So it looks like stem cells are there not to repopulate the endometrium in every menstrual cycle, but to repair in response to injury. Since one of the topics here was environment, I wanted to talk about one environmental influence that actually prevents recruitment of stem cells. Tobacco smoking seems to be one environmental agent that actually inhibits that recruitment. We see here recruitment of stem cells. NS means non-smoking exposed mice, and CS is mice that were exposed to cigarette smoke. And we looked at the number of stem cells recruited, and you can see in the epithelium in this particular study, or part of the study where we looked at cytokeratin positive cells, there are almost none recruited in this time duration that we gave them in the cigarette exposed mice. So there's certainly injury can recruit cells. There are toxins that may prevent recruitment of those cells and may explain some of the protective effects of cigarette smoke on endometriosis, for example. This is in endometriosis. Again, I've been talking about endometrium up to this point, but endometriosis, using our same mouse model we show in endometriosis, that stem cells here tagged in a couple different ways are recruited to the endometriosis. So this, we think, is a novel origin of endometriosis, that stem cells contribute to the disease. I think they account for the endometriosis that we see remote from the peritoneal cavity. And more than just endometriosis, this ectopic differentiation of stem cells is a new mechanism of disease, and endometriosis may be the first of what I wouldn't be surprised are other diseases that we start to uncover are caused by ectopic differentiation of stem cells. Well, endometriosis is very good at recruiting stem cells. The inflammatory environment, the cytokines that are produced recruit more stem cells in the uterus. The black bar here is endometriosis. The yellow bar is the uterus, so it outcompetes the uterus. When we look at the uterus in an animal where we've created endometriosis, you can see there are far fewer stem cells that are recruited to the uterus in the animal with endometriosis than the animal without endometriosis. We think that endometriosis is so effective at recruiting stem cells that it competes with the uterus for a limited supply of stem cells. It essentially outcompetes the uterus for a limited supply of circulating stem cells. And that's what we see in our treatment model. Here's a control animal. Here's a control animal where we're given endometriosis treatment and looking in their uterus, no effect. Here's the animal where we induce endometriosis and look in the uterus, far fewer stem cells recruited. When we treat the endometriosis and several different medical therapies work, we see a restoration of that flux of stem cells to the uterus. So endometriosis, we say, acts as a stem cell sponge. It soaks up that limited supply of stem cells and may allow better uterine repair in response to injury. What attracts these stem cells to the uterus in endometriosis? Well, to make a long story short, we've looked at different chemokines, and it's the CXCL12 that we found is the main chemoattractant. When we look at chemoattractive index of stem cells to CXCL12, we took here endometrial stromal cell conditioned media. You see about a 7-fold increase in recruitment towards the conditioned media, and we can block that in a dose-responsive fashion with AMD 3100, which is the CXCL12 receptor antagonist. So it looks like blocking that CXCL12 leads to or that CXCL12 leads to that recruitment. The other thing we found, though, is not only this traffic of stem cells to the endometrium and endometriosis, but actually stem cells reside in the endometriosis and can leave and go other places. We just published this in Stem Cells where we show this flux of cells out of the endometriosis. Here we used in a mouse model DS red cells as our tag to identify the transplanted endometrial cells, and we looked in the circulation and we sorted for this DS red marker. And what we found is these cells did not have markers of differentiated endometrial cells. They had all of these mesenchymal stem cell markers, not hematopoietic stem cell markers. These were actually stem cells in the circulation leaving the endometriosis. Where do they go? Well, the first place we looked, these stem cells can migrate back to the uterus. That's been shown in many metastatic tumors, that cells from the tumor migrate back to the primary lesion. And we find both by cell sorting, PCR, or here immunofluorescence for GFP, that these cells from endometriosis, and GFP tagged cells these are in our animal model of endometriosis, migrate back to the uterus. And here we show that they express some epithelial markers like Wnt7a once they get there. But what I want to point out here is, again, we make sure they're not CD45 leukocytes. They have, these are the GFP cells that made it back to the uterus. They express markers such as Wnt7a and cytokeratin epithelial markers. But look where these GFP expressing cells are. They're out here. They're not in the glands like the other epithelial cells. When they do come from outside from the endometriosis to the uterus, they are not incorporated normally in the endometrium, and that may interfere with cell-cell signaling, with gradients of various molecules that are present in the endometrium. The other thing we found is we started to look at other organs. Well, if we go to the uterus, where else does it go? Something we've called micrometastasis of endometriosis to different organs. We did the same thing here, DS-red positive cells. We created endometriosis in mice. This is our positive control, there's our negative control, and I'm not sure if you can see it from the back of the room, but in the endometriosis mice, we didn't see any in the controls, but in the endometriosis mice, you can see a few dots, not a lot, to the right of this line, meaning that in the lung, the spleen, the liver, the brain, there were a few cells from the endometriosis that ended up in these various organs. Again, these are very rare, but when we counted them, you can see there's some in spleen, more in brain, more in liver and lung. Although rare, extremely rare, we found these in every animal that we looked at. Now, what the clinical implication of these cells going there isn't clear, but they're not CD45 cells. These DS-red cells, again, found lung, spleen, liver, and brain, are not expressing CD45. They are incorporated in this tissue, and what that means, they're not forming endometriomas, but whether they're having an impact on these other organs, we haven't determined yet, but I'd like to suggest they probably do. A cartoon that's going to summarize this. The first and classic cell trafficking in endometriosis is retrograde menstruation. Cells leave the uterus. We also know that bone marrow can contribute to both uterine endometrium and a larger arrow, because endometriosis is actually very effective at recruiting these stem cells. Those cells can go back to the uterus, and some of the cells actually go and incorporate in other organs. The other thing I'd like to just briefly mention, because our whole next talk will be on microRNAs, so I'm not going to go into any detail on this, other than to say we've been studying microRNAs for a long time. Some of them are decreased in endometriosis. This is, say, the LET7 family of microRNAs. But they're also found in the circulation, where we've started to use them as potential biomarkers of endometriosis. And this is a paper we published a couple years ago. We see that several microRNAs are greatly increased in the circulation of women with endometriosis. Some are decreased. MicroRNAs are circulating around the body in endometriosis, and they're not just there for us to use them as biomarkers. They probably have a functional impact. So I think stem cell migration and circulating microRNAs both affect distant organs outside of the pelvis. Again, it's not just about the few lesions we see in the pelvis. This really is a systemic multi-organ disease. To prove that, we did some experiments here where we looked in the gene expression of some genes known to be involved in implantation in the uterus in animals where we created endometriosis in the pelvis, and we created endometriosis elsewhere. We put it between the shoulder blades, remote from the uterus. Most of the time, the uterine effects when the endometriosis was remote were just decreased in response to what we saw in the pelvis, but a few were actually more pronounced. So even remote endometriosis can have an impact on the uterus. These circulatory factors, whether it's the inflammatory state, whether it's the microRNAs, or whether it's cell migration, we really have to think again of endometriosis as a multi-organ system disease. Here's a paper we just published this year where we looked at some of the microRNAs that we found aberrantly expressed in endometriosis, and we treated human macrophages with either increasing microRNA 125, which is increased in women with endometriosis in the circulation, or decreasing LET7, which again is decreased in women with endometriosis. We saw increased expression of TNF-alpha, IL-1 beta, IL-6, IL-8, various inflammatory mediators that we know are altered in women with endometriosis. These microRNAs that are induced by the endometriosis are having a profound effect on the immune system. We've done other experiments. We started to look at endometriosis' effect on body weight, for example. Epidemiologic studies have shown that women with endometriosis are thinner. And I've heard it said, is being thin a risk factor for endometriosis? Well, in the animal model, we show no. The endometriosis is causative. We create endometriosis versus a sham surgery. The mice, the endometriosis, lose weight. It is caused by the endometriosis. Not only that, when we tried to explain that, we looked in their liver, and the genes like fibrin, LDH, albumin weren't changed, but a few genes that specifically are known to regulate metabolism were altered in these mice with endometriosis. We didn't see anything in their liver, but probably, again, through these remote signals communicating from the endometriosis to the liver, suggests that maybe there's a metabolic phenotype in endometriosis that we haven't really thought about yet, but probably has to do with communication from endometriosis to liver. We also looked in endometriosis mice to look at the effects on the brain. And when we transplanted endometriosis to mice, we saw changes in gene expression, electrophysiology, and behavioral changes in the mice. It's a little bit busy slide, but what I want to just summarize is this graph here shows some changes in gene expression, mostly in the hippocampus and amygdala of mice with endometriosis as opposed to those that had sham surgery. We look at the electrophysiology. Their electrical activity in the brain is different because of endometriosis. And when we do these behavioral tests, here we look at the mice motion around a cage, and mice with anxiety tend to hide in the corners and don't travel as much in the center of the cage. And you can see that in endometriosis, these mice had more anxiety. When we used a hot plate test, they eventually developed increased pain sensitization, so they're more sensitive to pain. And when we used the tail and mobility test, we showed that they had increased depression. So that many of the characteristics that we see associated with women with endometriosis really are caused by the endometriosis. And I find it frustrating. Sometimes I've heard some of staff say that endometriosis patients are difficult, they're anxious, and they exaggerate their pain. No, endometriosis causes anxiety, causes pain central sensitization, makes them more sensitive to this pain. So coming back to my original thesis, we know for example, the ASRM staging system doesn't really predict pain, doesn't predict fertility well. We know it's not just about what we see in the pelvis. I hope I've convinced you that this is truly a multi-organ systemic disease, not explained just by what we find in the pelvis. The endometriosis lesions that are seen in the pelvis are really just a small component of this disease. Circulating stem cells, circulating microRNAs, and probably other things that we haven't identified yet cause these systemic manifestations. And awareness of this systemic nature of endometriosis will allow for a better understanding of this pathophysiology and better understanding these clinical manifestations of the disease that we're all aware of. Just briefly, I want to talk to you about at least some of the developments that are coming along now in the future, what we can do based on this knowledge, treating these various other systemic conditions by blocking some of this signaling, or in this example, using stem cell transplant to improve pregnancy and Asherman syndrome. These are some of the original studies we did in mice a few years ago, where we first identified the ability of bone marrow stem cells to improve pregnancy rates in an Asherman's mouse model. So perhaps this may be helpful clinically, and there are some groups around the world that have already started using this clinically. But even better, I showed you earlier that CXCL12 is the chemokine that attracts these stem cells to the uterus. We just last year published this paper where instead of giving bone marrow transplant, we infuse CXCL12, inject it into the uterus, and you can see that it increases stem cell engraftment, increases litter size close back to the control here, and reduces time to conception. So again, improves infertility. So I'll end by just saying endometriosis is a chronic systemic disease, that stem cells and microRNAs are going to be useful in the development of new diagnostics and new therapeutics in the future, and just thank my collaborators and the rest of my group at Yale who've helped with this work. Questions?
Thomas Zollner from Berlin. You excellent and thought-stimulating talk. Thank you very much.
Thank you.
One very brief comment and one question. So I liked especially your aspect on anxiety and depression in your animal model.
Yes.
And actually we found exactly the same. So and the question is, did you ever, mesenchymal stem cells are known to work anti-inflammatory, and therefore they could be actually a treatment of endometriosis. Did you ever try in your animal model to transfect your lesions with SDF, with CXCL12, to treat endometriosis?
Well, not to transfect the lesions, but we're blocking that, and we haven't published that yet, but yes, we've got some data on that. It's effective.
And then it would worsen the symptoms of the disease, or?
If you, I mean, the SDF or the CXCL12 is used by endometriosis to recruit more stem cells and grows the endometriosis, so blocking it actually inhibits the growth of endometriosis. So in that way, actually beneficial.